Okay, thank you for the introduction. Thank you for giving me this chance to present my team's work here at this conference in my hometown of Ljubljana. This is a very fancy title for a very simple thing. I'm going to show you a case study of a drug discovery project. So we basically developed a new drug for treating symptoms of canine cognitive dysfunction. So if you give a dog suffering from this disease this drug, you improve the quality of its life and also its owner's life. But before I get into the details, I have to thank the institutions that made this research possible: the Faculty of Pharmacy, the Veterinary Faculty, the Faculty of Medicine, and also our partners from France and from Poland. Look, I'm not a smart man. If you don't believe me, you can ask my 9-year-old daughter. But I'm extremely lucky to get to work with these smart researchers. So everyone from Professor Gobec to Professor Majdic from the University of Ljubljana to everyone from Poland and from France. And of course, I also have to thank the money. I have to thank the agencies and the funds that have given us the money to do this research. So I'm going to start by being honest, right? We don't know what the cause of Alzheimer's disease is, right? What we do know is that we have spent a whole lot of money trying to answer this question. Develop all kinds of compounds that target inflammation, the amyloid cascade, tau protein, but the only answer we've gotten so far is these aren't the drugs you're looking for, right? But as long as we don't have the cure, we have to treat the symptoms. We have to help the patients by treating the symptoms. And I think we all know of a neurotransmitter in the brain called acetylcholine, which is responsible for memory, learning, and also attention. So we know this works. We know that if we increase the brain levels of acetylcholine, we're able to alleviate some of the cognitive decline, right? So if it ain't broke, don't fix it, and if it works, right? And we've been actually doing this for years now, using these drugs, galantamine, donepezil, and rivastigmine, which increase the brain levels of acetylcholine by inhibiting its enzymatic breakdown, by inhibiting acetylcholine esterases. And in the brain of healthy adults, acetylcholine esterase does most of the work, while butyrylcholine esterase plays only a supportive role. So from what I've told you so far, you would think that everything is okay, we don't need new drugs. Well, ain't that cute, but it's wrong. Because these drugs don't inhibit only acetylcholine esterase in the brain, but also in the peripheral nervous system. And they cause these adverse effects, which are very cleverly abbreviated by dumbbells. Okay? So that's one bad thing, but the other bad thing is that as Alzheimer's disease progresses, these drugs lose their efficacy. And researchers have found out that as Alzheimer's disease progresses, the enzymatic activity of acetylcholine esterase in the brain decreases, while the enzymatic activity of butyrylcholine esterase increases. So it would be smart to use selective butyrylcholine esterase inhibitors for treating symptoms of Alzheimer's disease. So a long time ago, in 2012, Professor Gobec called me into his office and told me that Boris discovered something very interesting. He took the crystal structure of human butyrylcholine esterase, the zinc compound database. This is a database of every compound you can purchase. And he also took into account structures and properties of known cholinesterase inhibitors, and he performed a virtual screening. And he then purchased 40 of the top scoring compounds, performed an in vitro enzyme test, and discovered this compound. This became our hit compound. It's a very potent and very selective inhibitor of butyrylcholine esterase. So if you want to know more about how Boris did what he did, please read this paper published in 2014. Okay, so whenever in a drug discovery project, when you get a hit compound, you want to see if you can improve the inhibitory potency by making some structural modifications. And you usually start this with the crystal structure of the hit compound bound in the active site of the target enzyme. Well, unfortunately, that was not available, so we started from basically just from the structure and made some logical modifications. So the boss wasn't happy when we did this change because we didn't improve the inhibitory potency. Then we changed this naphthalene ring with some benzyl rings. Again, the boss was not happy. But then the boss was happy when we made this change. When we changed this indene ring with benzyl groups, we made class 3 compounds and actually improved the inhibitory potency. We still wanted to see if we can go further. We changed this side chain with various smaller and different chains, but there was no improvement compared to class 3. And we also investigated these compounds, which are 1,4 desubstituted pyperidines compared to this 1,3. So we altogether we made 41 new compounds. We evaluated all of them, and this is our lead compound, which has improved inhibitory potency. So yes, Borat, great success. Okay, so I'm not going to bore you with the synthesis. I just want to show you how extremely easy it is. So we are able to make about 240 grams, starting from 100 grams of this amino acid in about seven days, which makes this synthesis very industry ready. So in the first step, we make this benzoyl amide. The next step, we make this D-amide. Then we reduce both amide groups. And this is the final step. We make the sulfonamide and then the chloride salt. And the great thing about this process is there is no purification of these intermediates necessary. So it's very easy. Yes, grumpy cat, it's that easy. It's so easy, a cave person can do it. Please, no offense to cave people, okay? Okay, so then we were very lucky when we obtained the crystal structure of this lead compound in the active site of human butyrylcholine esterase, which enabled us to explain the very nice inhibitory potency. So there is a strong cation-pi interaction here, there is T stacking, and there is a hydrogen bond. So this is not very common, so we were very happy when we got this crystal structure. But at the same time, we also got the crystal structure of the hit compound bound to the active site. And then we observed that they bind differently. And the major difference is here in the choline binding site. So this lead compound binds with its benzyl group, while the hit compound binds with this side chain. And this is, yes, Joey, that's awesome, because that's not typical. When you have a drug discovery project and you make a hit compound and then a lead compound, they usually bind in the same way, right? So that's very interesting. Okay, so this is just a short recap of the hit-to-lead optimization. We have the hit compound. We improve the inhibitory potency. So if the hit compound is this old Adam West Batman, then our lead compound is this Christian Bale Dark Knight Batman, right? So if you want to know more about how we did what we did, please read this paper again, published by Boris in 2014, and this one published in 2016. Okay, so before you proceed to any in vivo studies with any compound, you have to make sure it's selective, it's not toxic to cells, and it has a favorable pharmacokinetic profile. So we showed that our compound does not inhibit acetylcholine esterase, so no dumbbells. It's not cytotoxic to these two cell lines. It's highly permeable in CACO2 cells, which is very important when you consider oral administration, which is the choice of administration for drugs, right? It's highly plasma protein bound. It does get into the brain, which is imperative for compounds used to alleviate symptoms of Alzheimer's disease, right? And it's metabolically stable, so the half-life in human plasma is over 2 hours, and the half-life in human hepatocytes, it's close to 1 hour. So we then proceeded to cognitive investigation in vivo, and we chose the scopolamine-induced amnesia model, where scopolamine, when given to test animals, produces Alzheimer's disease-like symptoms, right? So what does scopolamine do? It's a muscarinic ACH receptor antagonist, which prevents acetylcholine from binding and produces memory and cognitive deficits, which are comparable to those observed in patients with Alzheimer's disease. Comparable. It's not the same, but it's comparable. So what do cholinesterase inhibitors do? They prevent the enzymatic breakdown of acetylcholine, which then displaces scopolamine from the receptor and restores normal memory and cognitive functions. So we performed 3 behavioral tests, and I'm only going to show you the results of one. The Morris-Watermaze test is a simple test used to investigate a compound's effect on long-term memory, cognition, and sensory motor functions. You take a pool with a hidden platform, and you put a mouse in the pool, and you measure the time it takes for it to swim and find this hidden platform, right? So on day 1, a healthy mouse or any mouse takes about 60 seconds to find this hidden platform. But then as days go by, as you train the mouse, it takes less and less time because it uses its memory, it learns where the platform is, and by day 6, it takes it only about 10 seconds, right? So that's a healthy mouse. If you treat a mouse with scopolamine, you produce Alzheimer's disease-like symptoms, you induce cognitive decline, and as you can see, it learns a lot slower than the healthy mouse. But mice treated with scopolamine and our compound, our sulfonamide, perform this test just as good as the healthy mice. So we can conclude that our compound has procognitive effects because it is able to reverse the scopolamine-induced cognitive dysfunction. Okay, so before we proceed to any trials in actual patients, you have to make sure your compound is safe. So drug safety first. And as I mentioned in the beginning, approved cholinesterase inhibitors cause dumbbells, right? So when you give them to rodents, they also inhibit acetylcholine esterase in the peripheral nervous system, and they produce memory impairment, salivation, urination, and defecation. Our compound is a selective butyrylcholinesterase inhibitor, which does not inhibit acetylcholine esterase, and it does not produce these adverse effects. So encouraged by these results, we struck a collaboration with Professor Majdic from the university, from the veterinary faculty, who actually told us that the dogs over the age of 11, about 50% of them, so every other dog, shows signs of dog Alzheimer's disease called canine cognitive dysfunction. And your dog may be suffering from this disease if he's getting lost in familiar places, has changed his sleeping patterns, has lost his appetite, doesn't want to play with you anymore, is acting aggressive, and has accidents inside the house, right? And if you would like to know, Professor Majdic talked about this yesterday for those who were at the lecture, but if you would like to know more, please read his paper published in 2019. Okay, so we were able to obtain 14 dogs with canine cognitive dysfunction, and we evaluated them, or the veterinarians evaluated them. They gave dog owners questionnaires, and they also performed 2 cognitive tests, and every dog got a score. And it's not like basketball when the more you score, the better you are. It's the higher the dog scores, the sicker the dog is, right? So we split the dogs into 2 groups, the control group and the treated group. And the dogs in the treated group were given our compound twice a day, orally for 6 months. And we evaluated the dogs before the trial, and after 3 and after 6 months. If you would like to know more about this study, please read this paper published in 2021. I'm just going to finish quickly with the results. So again, remember, the higher the score, the sicker the dog, and here are the results. So before the trial, all dogs were equally sick, right? They got similar scores, but after 3 months, the untreated dogs got sicker, and the treated dogs got better. They showed a drastic improvement in the symptoms. So we were very happy when dog owners reported that they experienced an enhanced quality of life for themselves and for the dogs. We have, of course, patented this compound, and if there are any representatives of pharmaceutical companies here in the crowd, please, you can wire me the money. Okay, so I would just like to finish with this. We have to look for the cure for Alzheimer's disease and other types of dementia, but please, let's not forget about what actually works, because there are a lot of people and a lot of dogs out there, and they need our help. Thank you for attention. Excuse me for exceeding the time a little bit, but please, any questions are more than welcome. Thank you.